Hello everyone, welcome to the class on amino acids biochemistry. In this video I am going to explain about amino acid classification, what are Zwitter ions, what do you mean by isoelectric pH, what is DL isomerism in amino acids, optical activity in amino acids, peptide bond formation, genetic diseases, and finally I will tell you how to easily memorize amino acid structures. So watch till the end. This is my channel. If you like the video content, do subscribe. Let's get into the topic. So amino acids, as the name indicates, it has got an amine group and it has got an carboxylic acid group. To denote that, they are known as amino acids. So amino acids are the organic molecules which contain an amine group and acid group. So the general structure of amino acid is this one. You depict an amine group, you depict an acid group, and you have a central carbon on which you have hydrogen and a side chain is there. So all amino acids differ only in the side chain. Now, if you see the structure, the carboxylic acid is the functional group and the carbon adjacent to carboxylic group is known as alpha carbon. So on this alpha carbon you have an amine group is there. So some of the textbooks call these amino acids as alpha amino acids because on alpha carbon you have amine group and they are acids. So they are also known as alpha amino acids. Both of them are same. Moving to the next one. See, as I told you, depending upon this R group, amino acids are classified. The R group could be of anything. Amino acids can have all group as polar groups, or they can have non-polar groups, or the R group can be aromatic ring, or R group can be acidic. In case of acidic, they are also known as negatively charged, or they can be basic. In case of basic, they are called as positively charged. Acidic means you have an additional COO group is there, hence it will get a negative charge, hence they are known as negatively charged. Basic means amine group is there which will get a positive charge, hence they are also called as positively charged. Now look at them. So this class is known as non-polar aliphatic R groups. All of them, this is a non-polar group. Non-polar means which could not generate any charge. No charge is there. Non-polar are also known as hydrophobic. They do not dissolve in water because they do not generate any charge. Glycine, alanine, proline, valine, leucine, isoleucine, methionine. All of them are considered as non-polar aliphatic R group containing amino acids. Whereas other one polar but uncharged amine groups are there. Polar serine, OH is there, threonine, OH is there, cysteine, SH is there, asparagine and glutamine, amide group is there. All of them are polar. So they are considered as polar uncharged R groups. In this group you have serine, threonine, cysteine, asparagine, glutamine. Now the other class is aromatic R groups. Phenylalanine, tyrosine, tryptophan. You can see all of them contains aromatic amino acids. Tryptophan has got an heterocyclic ring. Positively charged groups are also known as basic amino acids. As I explained you, the amine group contains a positive charge, hence they are considered as basic one. Lysine, arginine, histidine. Similarly, negatively charged group are also known as acidic amino acids. You have only two acids are there, aspartate, glutamate. They are also known as aspartic acid and glutamic acid. In ionized form, they are called as aspartate and glutamate. So this is how amino acids are classified. Moving to the next one, Zwitter ions. Zwitter ions are also known as hybrid ion or ampholyte. Hybrid means two. Zwitter means two ions. Why two? See, the amine contains a positive charge whereas the acid contains a negative charge. Hence they are considered as Zwitter ions, the positive and negative ion. So usually the non-ionic form of amino acid will be like this. Depending upon the pH, they will generate a kind of charge, and this double charge or hybrid ions is known as Zwitter ions. It has got a negative charge as well as a positive charge. It all depends upon pH. Now, moving to the importance of pH, there is something called as isoelectric pH. pH at which amino acids exist as Zwitter ions. Zwitter ions are neutral because it has got a negative and a positive charge, they will get neutralized. So the pH at which amino acids exist as Zwitter ion is called as isoelectric pH. Zwitter ions are neutral and they carry no net charge. Remember, it is not no charge, it is no net charge. Net means overall charge on the molecule. A positive and a negative balances and it becomes a neutral molecule. Now, the isoelectric pH value, see, this is what is isoelectric pH. You have low pH means acidic pH, H plus will be there, it will protonate an amine group and you will get a positive charge. And in high pH, in basic pH, OH will be there, that will abstract a proton from acid and you will get a negative charge. But at this pH, they exist exactly with a positive and negative charge, hence they are generating Zwitter ion. So this is called as isoelectric point. So both of them are related. Zwitter ions exist in isoelectric pH. Moving to the next one, DL isomerism. See, in the previous my video, I have explained about carbohydrate chemistry, in which we have seen L-glyceraldehyde and D-glyceraldehyde. So DL isomerism is about configuration at asymmetric carbon. Now, when you see glyceraldehyde, you have only one asymmetric carbon is there. The second carbon is asymmetric carbon. Asymmetric carbon is a carbon to which 4 different groups or atoms are attached to the carbon. Now, in asymmetric carbon, if hydroxy group present on the right side, it is considered as D isomer. If hydroxy group is present on the left side, it is known as L isomer. Similarly, in case of amino acids too, see, amino acids has got this one, asymmetric carbon, wherein you have 4 different groups are there. Take for the simple case of alanine. Now, with respect to carbohydrates, in case of amino acids, the amine group is considered. In carbohydrates, we have considered hydroxy group, whereas in amino acids, we will consider the presence of amine group. If it is on the right side, it is called as D isomers. If it is on the left side, it is known as L isomers. Now, the peculiar thing with these things is, in case of sugar molecules, all natural sugars are D isomers, whereas in case of amino acids, all natural amino acids are L isomers. Now, you can see the same structures. This is how the ball and stick model looks like. You have an alpha carbon on which, if amine group is present on the right side, it is a D isomer. If it is present on the left side, it is known as L isomer. So this is about DL isomerism. Now, amino acids will combine to form proteins. The functional units, the primary units of proteins are amino acids. Now, amino acids will combine to form peptide bond. How do they form? You have this one amino acid has got this carboxyl group, another amino acid, this amino group. In this, the hydroxy and this hydrogen comes out as water molecule, and this is known as peptide bond. This is nothing but an amide bond. Amide bond in amino acid is known as peptide bond. This is how all the amino acids will link together and form large chains of proteins. All amino acids will combine with this peptide bond. Now, this peptide bond can also be cleaved in presence of acid or base. This is what happens during our digestion. In presence of acid, all these peptide bonds will get cleaved and individual amino acids will get released. So this is about peptide bond. Moving to the next one. See, amino acids has got optical activity. See, optical activity is a property which denotes the ability to rotate plane polarized light. Some molecules will rotate the direction of this plane polarized light. If the direction is changed to the right side, they are known as dextrorotatory. If the direction is moved to the left side, they are known as levorotatory. Now, optical activity is related to chirality. Now, if a molecule is chiral, it will show optical activity. The relationship is absolute. That means if a molecule has got chirality, it will definitely show optical activity. Now, chirality is again related to asymmetric carbon. Most of the asymmetric carbon containing molecules are chiral. So, asymmetric carbon means the carbon should have 4 different groups attached to it. In simple representation, we write it as like this, 4 different groups. Then it will be a symmetric carbon and it will show optical activity. Now, there is only one exception. Only glycine is not optically active. The reason is glycine structure is this one. Look at this carbon. This carbon is not asymmetric. The reason is it has got 2 similar hydrogen atoms. If 2 similar groups are there, it won't be a asymmetric carbon. It needs to have 4 different groups or atoms. All other are optically active and they are chiral. Now, L amino acids are naturally occurring. I discussed this already. So this one is important. See, glycine is the only amino acid which is not optically active. There are 20 amino acids are there. Out of 20, only one is not optically active. Moving to the next one. Now, amino acids, depending upon the biosynthesis, they are classified as essential amino acids and non-essential amino acids. Now, essential amino acids, they cannot be created in body and they must be supplied from the diet. Whereas non-essential, human body can synthesize them. So, without these essential amino acids, the major function of the proteins will get hampered. So, they need to be supplied from the diet. There is a list of these amino acids. See, non-essential list is this one, essential list is this one. In some of my previous videos, I have given a mnemonic to memorize them. See, it can be remembered as PV, malt, hilt. Understand each and every one. See, P stands for phenylalanine, V stands for valine, M stands for methionine, A stands for arginine, L stands for leucine, T stands for threonine, H stands for histidine, I stands for isoleucine, L stands for lysine, and T stands for tryptophan. So if you remember this one, you will get the list of essential amino acids. Now, some of the textbooks will consider arginine as non-essential because arginine can be synthesized in human body. True, but the amount of arginine which is synthesized in human body is not enough to meet the metabolic demands. So what happens is, whatever the arginine is synthesized, it is consumed to make urea, a metabolic product of the amino acids. So arginine, whatever is biosynthesized, is utilized in converting urea, so the arginine levels are not enough, hence it is also considered as essential amino acids. Now, phenylalanine is used to synthesize tyrosine. Now, if phenylalanine is not supplied through the diet, people also have deficiency of tyrosine. So this thing you need to understand and remember. So this is the list of essential and non-essential amino acids. Right? Let's move to the further one. Now, there are some human genetic disorders which are affected by amino acid catabolism. The important diseases are albinism, Alkaptonuria, maple syrup urea disease, Methylmalonic Acidemia, Phenylketonuria. Now, see, in case of these diseases, what is important is you need to understand what is the defective process and what is the defective enzyme and what are the symptoms. Let us see one by one. Now, first one is phenylketonuria. First, understand this word, phenylketonuria. That means phenyl ketones. Phenyl ketones are present in urine. Hence, the condition is known as phenylketonuria. Why is this happening? See, phenylalanine is converted to tyrosine with the help of an enzyme, phenylalanine hydroxylase. You can see the difference is only hydroxyl group at this junction, and the hydroxyl group is introduced by phenylalanine hydroxylase. If this enzyme is not working, it results in phenylketonuria. Phenylketonuria is normally denoted as PKU. Now, what happens with that? Phenylalanine levels increases. So, one of the major sign of the phenylketonuria is phenylalaninemia. Phenylalaninemia. Emia means blood. Phenylalanine means phenylalanine amino acid. So, this amino acid concentration will be very high in the blood. Now, not only this, the normal metabolic pathway is phenylalanine will get converted to tyrosine. If this pathway is not working, what happens? Phenylalanine will undergo this kind of metabolic pathway, wherein phenylalanine is converted to phenyl pyruvate. What is the difference? The amine group is removed with the help of amine transferase. So this is phenyl pyruvate. Now phenyl pyruvate is converted to phenyl acetate and phenyl lactate. Now see, this phenyl pyruvate and phenyl lactate, these two levels increased in urine. What are these two? This is phenyl group and this is a ketone. The phenyl group and ketone. So they are known as phenyl ketones. So because of the presence of these phenyl ketones, it is known as phenylketonuria. So what are the signs? Phenylalaninemia, phenylketonuria. Now in fact, this phenyl acetate gives a characteristic musty odor, a different kind of smell to the urine. So most of the time in hospitals, doctors will realize when kids will have this kind of urine odor. So this is about phenylketonuria. But what is the problem with phenylketonuria? If phenylketonuria is not treated, it results in mental retardation. Mental retardation. That means intelligent quotients will severely reduced. That is what happens with phenylketonuria. So how it can be treated means, see, what is the problem? Phenylalanine. Phenylalanine metabolism is inhibited. So see, phenylalanine is an essential amino acid which has to be supplied through the diet. So dietary restriction of phenylalanine should be there. Very small amount of phenylalanine should be supplied, which is enough to convert into tyrosine. Any excess level will cause phenylketonuria. It has to be treated as early as possible, otherwise it results in mental retardation. Mental retardation. The IQ levels will severely affected, it will go down. So dietary restriction of phenylalanine is required. There is an artificial sweetener called as aspartame. Aspartame contains phenylalanine derivative, so it has to be reduced as much as possible. Otherwise, it increases the levels of phenylalanine, which increases phenylketones in the urine and causes this problem. Moving to the next genetic disorder, albinism. Albinism is again because of defective tyrosine catabolism. See, phenylalanine is converted to tyrosine. If the enzyme is defected, phenylketonuria will come. Tyrosine is converted to dopa. Dopa can be converted to dopamine and melanin. Melanin is because of this enzyme tyrosinase. Now melanin is a dark pigment which is present in our skin and eyes. Now the job of melanin is it will protect from sun rays. So it is required to protect our skins and eyes from the harmful radiation. Now in albinism there is a defect in this enzyme, so melanin will not form. If melanin is not formed, the dark pigmentation will not be there. So the major symptoms of albinism is white skin, white or pink skin, and white hair, completely white hair, because the dark pigment is not there. It also causes a condition called as photophobia. Photophobia means photo means sun rays, phobia means fear. They cannot face sun because it will give burning sensation. What is the job of melanin? It protects us from sun radiation. If that melanin is there, it causes burning sensation to the skin and eyes. It is called as photophobia. So this is about albinism. Again, what we need to remember, what is the defective enzyme, what are the signs and symptoms. Now, moving to the next one, Alkaptonuria. Now, tyrosine catabolism includes the conversion of gentisinate to mallyl esteoacetate. Now, the enzyme required is homogentisate 1,2-dioxygenase. If this enzyme is defective, it results in Alkaptonuria. In Alkaptonuria, there is a presence of dark pigment in the urine, and the dark pigment is nothing but homogentisate. Why? It is not getting metabolized, so it comes out from the urine, and this dark pigment is nothing but alkaptone, homogentisate. Now what is the problem with this? This may cause in late age, it results in arthritis. Arthra means joints, itis means inflammation. The pigment, the homogentisate may accumulate at the bone junctions and it may result in arthritis. So what is the enzyme responsible? Homogentisate 1,2-dioxygenase. What happens with that? Dark pigment will come in the urine, a black color urine, and at late stage it will result in arthritis. Now the next one, see, maple syrup urine disease is a somewhat problematic one. It occurs due to branched chain amino acid defective metabolism. Valine, isoleucine, leucine are called as branched chain amino acids. Now with the help of an enzyme aminotransferase, they form alpha keto acids. You know, all amino acids are considered as alpha amino acids. When this amino is transferred, you will get alpha ketones. So they are known as alpha keto acids. Now these alpha keto acids will undergo dehydrogenation and converted into coenzyme A forms. If this enzyme is defective, it results in maple syrup urine. See, the disease is called as such because the urine will have maple syrup color. Hence, it is known as maple syrup urine. But the major problem with this disease is it results in vomiting, it results in epilepsy or convulsions, and it may cause early stage death also. So this is a problematic disease. So it also results in mental retardation. So the major symptoms are, it causes vomiting, it results in convulsions or epilepsy, it results in mental retardation and early age death also. So maple syrup urine is majorly because of branched chain amino acid defective metabolism. Now the next one, methylmalonic acidemia. See, as the name indicates, methylmalonic acid accumulates in the blood and urine. Emia means blood. In the blood, this amino acid levels are increased. what happens is, see, all these amino acid isoleucine, methionine, threonine, valine will result propanyl CoA. The conversion of propanyl CoA to methylmalonyl CoA is inhibited, and that is what results in excessive levels of methylmalonic acid. And again, this also causes vomiting, convulsions, mental retardation, and finally early stage death. It may also cause early stage death. So this is also a kind of dangerous disease. So when you put all of them together, see, now understand this thing. Albinism. What is the defective process? Melanin synthesis from tyrosine is defective process. What is the enzyme? Tyrosine monooxygenase or tyrosinase is defective. What happens with that? Lack of pigmentation and white hair, pink skin. This is what happens. Now next one, Alkaptonuria. What is the defective process? Tyrosine degradation is defected. What is the enzyme involved? Homogentisate Y2 dioxygenase 1,2. Now, what are the symptoms? Dark pigment in urine, late development of arthritis. Now, the next one is maple syrup urine disease. This is also known as branched chain ketoaciduria. Now, defective process isoleucine, leucine, valine degradation is defected. Why? Because of the lack of dehydrogenase complex. What are the symptoms? Vomiting, convulsions, mental retardation, and early death. Now, what is the next one? Methylmalonic acidemia. What is the defective process? Conversion of propanyl CoA to succinyl CoA. This is inhibited, and methylmalonyl CoA mutase enzyme is defective enzyme. What happens with that? Vomiting, convulsions, mental retardation, and early death. This is dangerous one. Now, next, phenylketonuria. Phenylketonuria, you can see it is approximately it occurs approximately in high levels, abundance, this one and albinism. Both of them, the occurrence is very high. You can see 3 in 1 lakh incidences, whereas this one is 8 in 1 lakh births. So these two are in high incidences. Now, phenylketonuria, what is the defective process? Conversion of phenylalanine to tyrosine. What is the defective enzyme? Phenylalanine hydroxylase. What are the symptoms? Neonatal vomiting and mental retardation. So this list is important. Take a screenshot of this and understand these things. What is the defective process? What is the defective enzyme? What are the symptoms? Now, the last part, moving to the last one, learning amino acid structures. See, learning amino acids is fun. See, the simplest amino acid what we can see is glycine. See, simplest one, because you have an acid group, you have an amine group, but both of them carbon has got hydrogens. This one is known as glycine. Glycine, as I told you, it do not have optical activity. Now, after this glycine, the next important one is alanine. Alanine, the only difference is in this methyl group. See, this alanine is very important one. If you understand the alanine structure, based on this alanine, so many amino acids are there. Let me explain one by one. Look at this. See, alanine has got a methyl group. If to this methyl group, if you attach a hydroxy group, it becomes serine. This is serine. See, to alanine, attach one hydroxy group, you will get serine. Now, similarly, if in place of SH, if you have SH, a thiol group, it becomes cysteine. See, based on the alanine, we are developing all the structures. Alanine, you added OH group, you will get serine. You attach SH group, you will get cysteine. Now, along with that, you have one more amino acid is there called as phenylalanine. So, alanine is with methyl group. You attach a phenyl group, it becomes phenylalanine. So, again, this is also based on alanine. Now, on the fourth place, if you attach a hydroxy group, see, this is 1, 2, 3, 4. You attach a hydroxy group, you will get tyrosine. See, most of the inborn errors in amino acid metabolism, we are looking with this tyrosine. And this conversion is what affected in phenylketonuria. What is the enzyme? Hydroxylase, phenylalanine hydroxylase, because this hydroxy group is attached. So, attach a hydroxy group to tyrosine, you will get phenylalanine. So, all of them, serine, cysteine, phenylalanine, all of them are based on alanine. Not only this thing, you have acidic amino acid is there, aspartic acid or aspartate. To alanine, you attach an acid group, you will get, this is aspartic acid. A simple one, you will get this one, aspartic acid. So all of them, again I am repeating this. Remember this thing. You know alanine, you will get most of the structures. Serine, you remove a hydrogen and attach a hydroxy group. Cysteine, you attach a thiol group, right? And then phenylalanine, you attach a phenyl group. And then tyrosine, you attach a 4-hydroxy phenyl group. And finally aspartic acid. Aspartic acid, you add one more acid group. See, all of them are alanine derivatives. Now, leaving this, you need to know some basic amino acids. See, basic amino acids, we have lysine and arginine are there. Now, lysine, you have got, this is a butyl chain and then an amine group. So, this is known as basic amino acid because you have one more additional amine group is there. It can get a positive charge, hence it is also known as positively charged amino acid. Right. Now, leaving this, some other important things. There is something called as branched chain amino acids. You know, branched chain amino acid defect is what results in maple syrup urine. Branched chain amino acids. Now, look at the structures. See, for all amino acids, you need to write down this common formula. Only the difference is in R group. Now, if this R group is isopropyl group, so this is known as isopropyl group. This is known as valine. You have a branched chain, hence these are known as branched chain amino acids. Now, if you have this branched chain is isobutyl, so the same thing with isobutyl. This is known as leucine. Now the last one, if the branched chain instead of isobutyl, if you have a 2-butyl group, it is known as isoleucine. So this is known as isoleucine. Now look at them. See, valine, it has got an isopropyl group. Leucine, it has got an isobutyl group. This is, see, leucine and isoleucine are isomers. You can see the side chain is butyl one is isobutyl, and the one is 2-butyl. And this one is isopropyl. So all these three are known as branched chain amino acids which are involved in maple syrup urine disease. So see, learning amino acids is very easy. Again, the same classification, if you do practice by learning some of the amino acids, it will be always helpful. So thank you for watching this video. If you like this video, do subscribe.